In this video, I will be talking about the function of the kidneys. The kidneys work to maintain balance between osmolarity, water, and pH. To start off, we'll be looking at how the kidneys help to maintain homeostasis of pH. Homeostasis of body pH is maintained through an interaction of buffers, respiration, and the kidneys. Respiration through carbon dioxide, and the kidneys through regulating the amount of bicarbonate in the system. First, let's review the anatomy of the kidney. I'll be drawing it very simplified, but it will give you a basic idea of what's going on. The ureter leading to the bladder leaves the kidney from the major and minor calyxes. The renal pyramids lead into the minor calyxes, and the renal pyramids are part of the renal medulla. The capsule surrounds the kidney on the outside, and the part in between the capsule and the medulla is the cortex. The capsule also juts down in between some of the renal pyramids. Taking a closer look at the renal pyramids, structures called nephrons are present. These structures are responsible for the reabsorption of bicarbonate. Nephrons contain small capillary blood vessels called the glomerulus, which filters the blood into the urine, traveling through the rest of the nephron tubules into the collecting duct. The capsule, or top part of the nephron, is in the cortex, and the bottom part is contained in the renal medulla, or pyramid. So I actually drew the little bracket wrong before I drew the close-up of the pyramid. Some of the brackets should be going into the cortex. Just a reminder of the different parts of the nephron so that we are on the same page as we refer to some of these structures later. The afferent arteriole is bringing the blood. The efferent arteriole is taking the filtered blood away. Bowman's capsule contains the glomerulus. The proximal tubule is the part connected closest to Bowman's capsule. Capillaries will follow the tubules to exchange nutrients with the fluid within the tubules. Then there is the loop of Henle and the distal tubule, which will dump into the collecting duct, which will then dump into the minor calyx, and the urine will make its way out of the kidney. Four processes take place at this level: filtration, reabsorption, secretion, and excretion. Filtration occurs at the glomerulus. Reabsorption happens at the proximal tubule, the thin and thick parts of the loop of Henle, the distal tubule, and the collecting duct. Secretion occurs at the proximal tubule, the distal tubule, and the collecting duct. And excretion is the word for anything left in the collecting duct after the other processes take place and the urine leaves. So excretion equals filtration minus reabsorption plus secretion. Now we are going to look at a few of these sites that participate in bicarbonate reabsorption. These sites are in the proximal tubule, the thick loop of Henle, and the collecting duct. I'll draw a diagram of how bicarbonate reabsorption occurs across the cell barrier between the lumen of these parts of the nephron tubule and the renal interstitial fluid, or we can think of this fluid as the blood because nutrients here will eventually go back to the blood. So sodium and bicarbonate are present in the tubular lumen. Using a sodium-hydrogen antiporter, sodium enters the cell and hydrogen enters the lumen. Hydrogen and bicarbonate together form carbonic acid, which then can split into carbon dioxide and water. Carbon dioxide then uses simple diffusion to enter the cell. Carbonic anhydrase uses CO2 and H2 to again form carbonic acid, which can split into bicarbonate and hydrogen ion. The bicarbonate can then leave the cell by simple diffusion. And notice how one side of the cell is permeable to bicarbonate and the other is not. So levels of sodium in the cell are maintained by a sodium-potassium ATPase. Most of the bicarbonate reabsorption occurs in the proximal tubule. During acidosis, the kidney will reabsorb all the bicarbonate. In contrast, during alkalosis, the kidney will reabsorb less bicarbonate. Okay, now we are going to look at how the kidney synthesizes new bicarbonate and uses phosphate buffers, after which we will look at acid secretion. Hydrogen phosphate enters the urine by filtration at the glomerulus. About 90% of the filtered ion is directly reabsorbed. However, the phosphate ions that remain in the urine play an important role in acid-base balance. Hydrogen leaves the cell and joins hydrogen phosphate in the tubular lumen through a sodium-hydrogen antiport. This hydrogen comes from a reaction inside the cell forming bicarbonate. It is the same reaction that we saw before. This process occurs in all parts of the kidney, but most importantly in the distal tubule and collecting duct. It results in one hydrogen ion and one bicarbonate. Now we are going to talk about ammonia buffers. This process occurs in the collecting duct. It involves the direct active pumping of hydrogen, which combines with ammonia in the urine. One new bicarbonate ion is synthesized in this process. So you've seen a lot of these processes before. I don't really need to talk you through them in a lot of detail. So ammonia is passively leaving the cell and going into the tubular lumen. And then here you'll see the hydrogen will leave and it will join with ammonia. To end, we'll glance at the primary active secretion of hydrogen into the tubular lumen. This process is very similar to the other processes we've seen, but this time we won't include any buffers. This occurs in the late distal tubule and the collecting duct, utilizing a hydrogen pump. It increases greatly the hydrogen concentration of the urine, decreasing the pH, which is why the urine is normally acidic. The lowest urine pH is about 4.5. It can pump against a 900-fold gradient, which is crazy. In this process, a new bicarbonate is also synthesized. Watch the next video to learn about more processes important to this acid-base balance.